Hi innovation students, and welcome to the final lecture that will conclude the section on CRISPR. In the previous lecture, we described how the CRISPR system was discovered and how it works as an adaptive immune system in bacteria. We will now start discussing how CRISPR has been modified and adapted as a tool for genome editing in eukaryotic cells. We will also describe some of the applications of CRISPR-Cas technology. Now that we understand how the CRISPR-Cas system works in adaptive immunity, any sequence that contains a PAM or an NGG sequence could be targeted for cleavage in vitro as long as we had two components. That's an sgRNA and a Cas9 enzyme. The sgRNA is a customized RNA that contains the crRNA or CRISPR RNA as well as a tracer RNA-derived sequence. Plasmids can be engineered as expression vectors that carry genes which encode the Cas9 enzyme or an sgRNA, and that sgRNA can be modified to contain a specific DNA sequence that we would like to target, and this can be introduced into mammalian cells in culture. The CRISPR-Cas9 system will then cut a very precise sequence in the genome and form a double-strand break. When a double-strand break is formed in the genome, eukaryotic cells have endogenous DNA repair mechanisms in order to fix that double-strand break. However, these mechanisms can be error-prone. For example, non-homologous end joining can result in the insertion or deletion of nucleotides at the site of the double-strand break during repair. Another mechanism for the repair of double-strand breaks is homology-directed repair. Homology-directed repair specifically edits a double-strand break correctly by sister strand invasion. So when there is a double-strand break, the homology-directed repair system directs that region of DNA to a sister chromatid or homologous region of DNA and repairs the double-strand break by copying nucleotides from that sequence. Homology-directed repair can be utilized during genome editing, as we can use the system in order to introduce specific edits through sister strand invasion. Now let's talk about how this actually works by looking at this diagram. We have two expression vectors here, Cas9 and sgRNA. The Cas9 expression vector would contain a promoter as well as the sequence encoding the Cas9 enzyme. The sgRNA expression vector or plasmid would contain a promoter, a 20 nucleotide targeting sequence that's specific to the gene or the region in the genome that we would like to introduce the targeted genome edit, as well as the tracer RNA that will allow this region to bind to the Cas9 protein. In order to introduce targeted genome edits, a donor template can also be included in this reaction. So now, in mammalian cell culture, we'd have expression of the Cas9 enzyme, as well as a guide RNA sequence that will contain a CRISPR RNA or 20 nucleotide targeting sequence that will allow Cas9 to be guided to a specific region in the genome that we would like to edit. Once the Cas9 enzyme comes into contact with this region, it can then cleave that DNA at a very specific point upstream of the PAM motif. During non-homologous end joining, this double-strand break will then be repaired. As NHEJ is an error-prone mechanism, this may result in an insertion or deletion mutation at the site of the double-strand break. This could create a frame shift mutation, which results in the production of a mutant protein product. And so we could use CRISPR to modify a gene by introducing an indel mutation, and that would result in the formation of a truncated protein or a non-functional protein product. We could then use the CRISPR-Cas9 system in this instance for knockout studies. So if we wanted to investigate the function of a protein, we could use CRISPR-Cas9 to knock out the function of that protein and then observe the effects on mammalian cells in culture. Now, if we were to look at the HDR mechanism, during this mechanism, we would introduce a donor template as well as the Cas9 and sgRNA expression vectors into a mammalian cell. The donor template contains the region that has been cleaved, as well as a desired genome edit in the sequence. Now, as I've mentioned, during homology-directed repair, the double-strand break is repaired through sister strand invasion. Now, if we were to introduce a donor template that has sequence homology to the regions of the gene on either side of the double-strand break that we would like to introduce, rather than invading the sister chromatid, the HDR mechanism would use the donor template in order to copy nucleotides to repair the double-strand break. When it binds to the template DNA sequence rather than the sister chromatid, it would copy the DNA sequence along with the edited region that we have artificially introduced into the donor template. And this would result in a specific edit to a gene that we have directed the Cas9 enzyme to. And this is how the CRISPR-Cas9 system has been modified for use in genome engineering. Now, as mentioned, non-homologous end joining results in the ligation of broken DNA fragments. And due to the fact that non-homologous end joining is error-prone, this results in small insertions or deletions at the repair site. Homology-directed repair uses an undamaged homologous chromosome or sister chromatid as the template to correctly repair a broken chromosome. In terms of using homology-directed repair to our advantage, this system can be tricked into using an artificial donor template instead of the homologous chromosome in order to make complex substitutions, deletions, or additions to a gene. The homology-directed repair system is a less error-prone system, and so we can really utilize this to our advantage to create targeted genome edits in vitro. Although, when we're using the CRISPR-Cas9 system for molecular biology, we have to bear in mind that this system also has infidelity. Cas9 may not always cut the intended DNA target, and it can have off-target effects in the genome, which means that it can cut other regions of the genome, and this may be due to the sgRNA having more than one perfect match. Remember, the sgRNA has a 20 nucleotide targeting sequence, and although it's quite specific, it may potentially have off-target effects. And so we need to improve the specificity of these CRISPR-Cas9 edits in order to ensure that this technique is safe and so that it can be used in medical applications as well. CRISPR-Cas technology has diverse applications. The system has been modified for use in basic genetic research, which we will be discussing further. It can also be used in biotechnology for the cost-effective production of GMOs, genetically modified crops, that have enhanced nutritional value, increased pest resistance, as well as drought resistance. CRISPR-Cas may also have clinical uses for the treatment of genetic disorders that involve single gene mutations. However, its use in gene therapy must be carefully considered and assessed for off-target effects before this technology is properly applied in a clinical setting. Our ability to efficiently and quickly delete a gene from the genome has only recently become possible due to CRISPR-Cas technology. Reverse genetics is a good way to learn about the function of a gene. And reverse genetics involves deleting the gene and then observing the consequences of this effect. And so CRISPR becomes a very useful tool in basic genetic research in order to investigate the function of these genes. We'll now discuss how CRISPR has been used in basic genetic research. Mutations to the HNH and RUVC nuclease domains can create a catalytically dead version of Cas9. So if you remember from the previous lecture, the HNH and RUVC domains were responsible for introducing a double-strand break in the targeted region of the genome. However, if these regions were mutated on the Cas9 enzyme, the Cas9 enzyme would be able to bind to the target DNA sequence, but it would not cut that target DNA sequence. This catalytically dead version of Cas9 is called dCas9. dCas9 has been modified in multiple ways. This enzyme has been fused to an activation domain, and this results in the activation of transcription of any gene, because now we can fuse the activation domain of a transcriptional activated to dCas9. dCas9 will then bind to the targeted region of DNA, and this would result in activation of that particular gene. In the same way, dCas9 can be fused to a repression domain. This transcriptional repressor can then be targeted to a specific gene, and this will allow the researcher to turn off any targeted gene as they wish to do so using dCas9. dCas9 has also been modified to contain a catalytic domain. This catalytic domain can also contain an epigenetic modifier enzyme, for example, a histone deacetylase or a DNA methyltransferase. So when dCas9 comes into contact with a gene or a targeted gene, and that dCas9 contains a catalytic domain, this will allow the researcher to add or remove epigenetic tags to that specific region. Additionally, dCas9 can also be modified to attach fluorescent proteins, such as green fluorescent protein. These enable the researcher to see where a specific sequence is located within the cell using fluorescence microscopy, and this can be modified for the use of RNAs as well as DNA sequences. This diagram shows how dCas9 has been modified in multiple ways to contain an activation domain of an enzyme, a repression domain, an epigenetic modifier, as well as a fluorescent protein. It will then bind to the region of the genome that we would like to target, bringing these domains in close proximity to the target DNA fragment, and thus inducing these epigenetic changes or marks to a very specific locus in the genome. And so dCas9, as well as the CRISPR-Cas system, has now been modified to be used as a very useful tool in basic genetic research as well as targeted genome editing. Thank you. That concludes this lecture.